we are provided with two biological sequences and we want to align them. Now, what are different techniques that are available for this alignment? Broadly speaking, we can categorize these techniques in two domains. These are global techniques and the other one is local techniques. So what are global alignment techniques? ग्लोबल का मतलब ये होता है कि जो पूरे के पूरे सीक्वेंस को काउंट करे, यानी कि हम क्या करेंगे कि कंप्लीट सीक्वेंसेस को अलाइन करने की ट्राई करेंगे. ऑन द अदर हैंड, इन लोकल अलाइनमेंट्स, इंस्टेड ऑफ वर्किंग ऑन द कंप्लीट सीक्वेंसेस वी मेक सब सीक्वेंसेस विद इन अ सीक्वेंस. और उसके बाद हम ये देखते हैं कि कौन से कॉमन सब सीक्वेंसेस हैं जो बिटवीन टू सीक्वेंसेस अलाइन किए जा सकते हैं. अगर इसे मैं पिक्टोरियली डिफाइन करूं तो वो कुछ इस तरह की फिगर होगी. जो फर्स्ट वन हमने यहाँ पे शो की है दैट इज ग्लोबल टेक्निक्स. इन ग्लोबल द रेड वन इज फर्स्ट सीक्वेंस एंड द ब्लू वन इज द सेकंड सीक्वेंस. और यहाँ पर हम कंप्लीट सीक्वेंस की ट्राई कर रहे हैं अलाइन करने के लिए, विच इज शोन बाय दीज लाइंस. जबकि लोकल में क्या हो रहा है? लोकल में ये है कि हम ये देखते हैं कि कुछ पार्ट जो है हमारा सीक्वेंस वन का कंपेरेबल है कुछ पार्ट के सेकंड वन के. सिमिलरली जो हमने सेकंड फिगर यहाँ पे लोकल से रेलिवेंट दिखाई है उसमें ये कोशिश की गई है कि ज़रूरी नहीं है कि वो एक ही पॉइंट पर हो. जैसे लोकल का जो लास्ट पार्ट है दैट कॉरेस्पॉन्ड्स टू फर्स्ट पार्ट ऑफ द सेकंड सीक्वेंस पार्ट एंड इट कैन बी रिवर्स ऑल्सो. What is global alignment now? The definition of global alignment is basically it makes an effort to align the entire sequences. And what happens? It finds best match of both sequences in their entirely. Again, the definition is simple. It takes both the sequences and then try to find a match using both sequences, and that is also for entire length. What is input? Of course, input is two sequences without alignment. And then what is the goal? Goal is to identify complete regions which are similar in both the sequences. And how they are similar or how they are useful? They are similar or useful, or it works when both the sequences are in similar length. It can be that there are some gaps, but they should be somehow similar in length. What are different applications? Applications can be in DNA alignment or in protein alignment. For example, it can help if we have two genes with same function, such as humans versus mouse genomes, and then comparing two proteins with similar functions. So first one is with respect to genes and the other one is with respect to proteins. Now local alignment. Now what is local alignment? As I said earlier, that local alignment tries to find a match between subsequences of the given sequence. What are the sequences or what are the input? Input is again two sequences which are not aligned, or they may not be related or not related. Then what is the goal? Goal is to find substrings within a sequence that can be matched. Now applications. It has more applications as compared to global alignment. Why? Because it will also identify some useful patterns that may not be available in the entire sequence, but they may be available in shorter segments also. It is again useful for finding proteins, DNAs, RNA sequences. So here are some different purposes or usefulness of local alignments. More meaningful. More meaningful. How it is more meaningful? Because it finds out conserved regions between two sequences. Conserved regions means that are similar basically, or they are meaningful. The other ones is they align different sequences which may be of different length also. For the first one, for the global one, we need sequences which are roughly in same lengthwise. But in local alignment, it can be different also. Again, it can align two partially overlapping sequences because it is working on subsequences. And the final is that it aligns two sequences where one is a subsequence of another. So what happens is if there is a one big sequence and the other one is a small subsequence, then it will give 100% similarity between these two sequences.